my hypothesis is that because the activity changes, and the pigs that were restricted, they seem to be moving much more than the pigs that were not fed restricted.
Welcome, everybody, to today's episode of the Swine at Canada podcast. My name is Dan Columbus, and I am going to be your host for today's episode. And with me today, I have Dr. Elda Dervishi, who is a research project manager at the University of Alberta. So thank you for joining us today.
Yeah, thank you, Dan. Thanks for having me.
Looking forward to today's topic. But before we get into that, all things behavior and feed restriction and everything, just because some people might not be aware of who you are and what you do, I'll just ask to maybe give a little bit of history on yourself and your journey so far.
Sure. I moved to Canada in 2011 in Edmonton. So I did I was born and raised in Albania, in Tirana, and then I did my undergraduate studies at the Agriculture University of Tirana, and from there I continued with my career doing my master and my PhD in Spain. There is where I worked with small ruminants in nutrigenomics, and then in 2011 I moved in Edmonton as a postdoc fellow. And I moved in here for 1 year, and I'm still here. And during this time, I've been involved in many research projects involving different livestock species like cows, pigs, dairy cows, and beef as well.
Working with nature and not against it, piglets fed AX3 see significant and improved feed efficiency. Producers know the reality of needing to reduce antibiotic and zinc use. AX3 Digest is a highly digestible novel protein that promotes improved in-barn performance, piglet health, and minimizes the need for zinc in the diet. For more information, visit www.protecta.com. That's www.protecta.com.
So well-rounded experience. You know, we don't get many people that have done that many species. I'd say even lived that many places. So it's
Yeah, thank you. It's been great so far. Actually, working in different species give you an enriched experience, and you can learn from one to the other and apply different approaches.
Yeah, I agree. It's one of the things that I tell my students all the time, to make sure that they talk to other students and people that are not necessarily working with pigs, because you can learn a lot from what they're doing as well and apply it. I think too often we get stuck in our species, right?
Right.
Maybe that's the way it's going to be. But because this is the Swine It podcast, we are going to talk about pigs today. And I know you've mentioned that you've been doing work with feed restriction in pigs and what effects that has on multiple aspects. So I will turn the floor over to you, and you can start letting the audience know what you've been doing and what you found.
Yeah, thanks again for this opportunity, Dan. It's great to share our experience and our results with the audience that is interested in swine. And specifically, this project, one of our project, explored the effect of feed restriction on the behavior, some aspects of behavior in finisher pigs. And the main topic here was investigate the relationship between a low feed restriction and the tail biting and some other damaging behaviors like skin lesions. So in this project, what we did was apply a low level of feed restriction, 20% of feed restriction in pigs entering the finisher stage. And this feed restriction level was applied for 3 weeks, during 3 weeks, at age 9, 10, and 11 of weeks of age. And so we had roughly 132 pigs in this experiment, and the experiment lasted from week 8 of life till week 20 of life. And what we observed there was that the feed restriction, this level of feed restriction, overall didn't impact the number of the skin lesions overall. So, but we observed that with the time going by, the number of the skin lesion would increase. And the interesting finding was that we found an interaction between the diet, the restriction, and the number of skin lesions in females. So what we observed is that females that were feed restricted, they have more skin lesions compared to the males during those 3 weeks of feed restriction. Also, an interesting finding was that female, independently from the feed restriction, females compared to males, they were having lower serotonin levels compared to the males. So that was an interesting finding. A main objective of this project was developing strategies to reduce the incidence of the tail biting. So what we know that tail biting is a problem in pigs because it causes damages, and also it increases some costs, no, if it is severe. So in our study, what we found is that this level of feed restriction actually reduced the incidence of the tail biting. So when I say the incidence of tail biting, I mean that we observed each video recording, and we counted, we observed each pig, and we counted how many times a pig performed the tail biting behavior and how many times a pig received the tail biting behavior. So we wanted to see and investigate the association between the feed restriction and the number of tail biting performed. And to our surprise, because our hypothesis actually was that the restriction is going to increase the number of the tail biting. However, what we found was that it decreases this level of the 20%, no? It decreases the number of the tail biting. And so this was a surprise. However, we also observed a small difference in the weight of the animals. And we know that weight is a very important trait, and the average daily gain, they are important traits for the industry, no? Basically, that's what they get paid for. So this project actually opens the door for other ideas, no? So maybe in the future, we could explore levels of feed restriction that they effectively reduce the number of the tail biting performed and maintains the growth. For example, study or explore the effect of a 15% or 10% of feed restriction, and see if we get both of those benefits, no?
Yeah, I was going to ask about how the performance was affected by the restriction, because obviously less feed is going to mean a restriction. So I guess the other thing is pigs are generally fed pretty close to the requirements as it is, So do you think that there's going to be any kind of level of restriction that is going to be, I guess, not have a negative effect on their performance, or are you looking at maybe like it's a trade-off that you have a slight drop in performance, but the benefits of not having the tail biters, kind of provides an economic benefit?
Yeah. So the idea is to find a level of restriction that is optimal. No, that it's not going to affect the ADG, the average daily gain, or the weight, the final weight, and it's going to reduce the incidence of these behaviors. So we usually think it's a trade-off. And sometimes, quite often, it is a trade-off, but not necessarily. It's just a matter of keep researching and investigating until we find that level that is beneficial for a wide variety of traits. No, because in this research, what we did, we focused mostly on the tail biting, skin lesions, focusing mostly on the welfare side of it. And we also checked the production of it. However, we didn't follow up with, for example, carcass trait. No, that would be very interesting to see that. What we saw in our research is that, yes, the ADG of restricted animals, it goes down for those 3 weeks. However, after that, we see a compensatory response. No, so the pigs that were feed restricted, they catched up in terms of the ADG. No, and at some point they even surpassed the full diet. The question is, if these pigs that catched up, the feed restricted pigs, how does that translate to carcass trait? Are they depositing more fat? Are they depositing more muscle? So we don't know that.
Yeah, it would be interesting to see, because maybe where your restriction is happening is where restriction has been shown to be beneficial for compensatory gain, right? And there are periods where restriction the pigs won't recover from. So it'll be interesting to see what you find when you start looking at this further. I think, like you said, the interesting finding, though, was that the restriction caused a reduction in the tail biting, which, I mean, yeah, in my head, it's like, well, if they're hungrier, you think that they're going to be looking for something to go on. So what is there a hypothesis or what you think, like why you saw the opposite of what you kind of expected?
Yeah. In many studies, it has been reported the contrary, what we found, no, especially in females, fed restricted females, but it has been a bit more restrictive. No, the level has been higher. They've seen that when the females are fed restricted, they see more vulva biting, behavior. We did in the finisher pigs. We found the contrary, and so beside the tail biting behavior, we did investigated many other activities, pigs activities. So the pen virtually was divided in several areas, and we studied the frequency of the visits, in different area of the pen, how much time they spent in different areas of the pen, how long they were active and inactive, and so on. So what we found was that the pigs that were restricted, they were more active. Okay, and this activity also, it is different from the morning and from the afternoon. What we find is that the pigs are more active in the afternoon compared to the AM hours. And so they spend more times being active and moving in different areas of the pen. For example, what we observed was quite interesting, that the pigs, they would spend a lot of time close to the feeding areas, to the where the feeding areas are located. So they'll move around there. And also when the pigs were feed restricted, they also go and visit more the feeding areas or water areas as well. So my hypothesis is that because the activity changes, and the pigs that were restricted, they seem to be moving much more than the pigs that were not fed restricted. And from our observation, the explanation is that because they're moving so much around the pen, so they're not interacting with each other. So tail biting usually happens when the pigs are laying down, they are quiet, and then they start chewing each other's tail. And this is my explanation right now. It's because they are so active and moving, maybe looking for motivation for the feed. So they spend less time interacting with each other. So this might be an explanation for this level of feed restriction decreasing the tail biting.
Yeah, I was going to say, it sounds like they're too busy looking for food to worry about their pen mates, right? Like,
yes, yeah.
Very interesting. Now, I know you said earlier that you videoed and you watched the interactions and that, which I don't envy behavior scientists at all when I see them watching videos, hours and hours of video interactions. And so, did you just look at, if a tail was bitten and if somebody was biting, or did you look at like which pig was doing the biting and which one was getting bitten? And was there any kind of like correlation or findings along those lines?
So, yeah, we did those observations and we did do it at the animal level, So we knew who was biting, which pig was biting who. And which pig was receiving, because the pigs were identified. There, it was interesting to find that, especially with the morning and the afternoon recordings, we saw such a difference. So in their activity, and a pig that was biting in the morning wasn't necessarily biting in the afternoon always, So the classification would change. The behavior is such a dynamic trait, So it's not a static variable. So yeah, we found that the pigs that perform the tail biting in the morning not necessarily are biters or performing tail biting in the afternoon. Yeah.
Too bad. You can't just take out the problem pig and then get rid of it.
Another idea of this project was, can we find, so the pigs were recorded for 9 weeks, once a week. And the other idea was, can we find a behavior pattern that is in common for the pigs that perform tail biting or those that don't perform but they receive, And unfortunately, we didn't find a pattern that would distinguish all of them. But what we found is that at certain weeks, for example, at week 16, 18, and 20 of life, we observe that tail biters, those pigs that will perform the tail biting, independently when in the morning or in the afternoon, they would visit more the water area. So that was an interesting finding, that these pigs, they kind of prefer going to a certain water area, and not both of them, because a pen is equipped with 2 different water areas and feeder areas, but the tail biters, they preferred going to a water source. So yeah, that was interesting too.
So maybe it's related to water availability and not feed.
So yeah, it might be.
So you mentioned, looking at like level of restriction. Are there other concepts that you're looking or planning to look at when it comes to tail biting that could potentially be a fix for this problem?
Oh, there are so many. So, yeah, that's a good question. Very often we focus only on one aspect of it, but we have to keep in mind that this behavior, it is affected by many factors. It's a multifaceted behavior, no? So to start with, nutrition is one of those, no? Or and we tackled this aspect, but also we have to keep in mind, for example, we can reduce the tail biting by using enrichment, no? It has been already studied and reported that providing enrichment for pigs, it's beneficial, for many aspects. It has to, however, not all the enrichments are going to reduce the tail biting. The other aspect of reducing tail biting, of course, is the density of the animals. How many pigs we're putting in a pen? Are they overcrowded? Obviously, if the pen is overcrowded, it's going to affect. Another aspect is the light regime. How much light you're providing to the pigs and how much darkness are you providing to the pigs. So those are just few of those other aspects that can affect the behavior.
Yeah. Lots of different things that that people have looked at. It's one of those problems that probably never be solved, but hopefully we can reduce it with some aspects. Yeah, so it'd be interesting. When you've done some of the other work, we'll have to have you back on to discuss some of those findings too. So I guess changing gears a little bit, because I know there's another topic that you work on that I think might be interesting, and that is your work with metabolomics and pig resilience, right? So let's maybe spend the next last third of the podcast. We'll talk about that. So what have you been doing in that area?
Oh, right, yeah, that's another area that our group, Livestock Gentech, has been working at, looking at the pig resilience and combining this with the omics technology. So there is an interest of having resilience pigs, from the industry, the entire industry. So we want to have those pigs that, even when they are challenged by disease, they are able to recover pretty quick. So this, in particular, this project was a huge project in collaboration with many institutions, few countries, mainly Canada and US. And so I was mostly focused on the metabolomics part of it. So we were interested in finding if we were able to find metabolites or markers that could be indicators of the pig resilience, And in this project, what we did was estimating different genetic parameters of metabolites in pigs and estimate genetic correlations between metabolites and different health traits or production traits. And we found those results are published already. We have found many interesting results in the metabolomics part. I would say that there's no magic or a silver bullet. Say, do this and you're going to have a resilient pig, or select for this and the pig's going to be resilient. So no, we didn't find that, but we did find many interesting results which can be applicable, especially on the selection part of it. If I remember properly, one of the interesting findings that we had was the genetic correlation between isobutyric acid and the residual feed intake. It was one of those interesting results that we found. Also we found many metabolites to be heritable. These molecules, they are heritable, and the highest heritability was found in glutamic acid. So this amino acid is highly heritable in pigs, and also different genetic correlations between metabolites and blood cells in pigs, which might give us an idea how these molecules, small molecules or metabolites, they are affecting, genetically affecting the composition of blood traits. So, this was another project that I was involved in. And for anybody interested in our work, or they can find the articles that are already published, or send me an email. I'll be happy.
It's interesting. I guess it should be so unexpected that some metabolites might be heritable, because they are going to be related to metabolism and probably efficiency of utilization. Personally, I think the glutamic acid one is very interesting, because that suggests to me that when it comes to protein efficiency, that there's something going on there, and maybe we can pick that up with that, because it is a major nitrogen shuttler across around the body, right? So, I guess when it comes to that, the question that kind of came to mind when you're talking about, you're relating this to resiliency, and I think, depending on who you ask, resilience is going to be different, how they define it. So I guess, how would you define it, or how was it defined for the purposes of this project?
Right. For the purpose of this project, what we did, we have a big number of pigs, like almost 4,000 pigs in the experiment. So how it was defined was the pigs were considered resilience, those pigs that had the best or the higher growth rate and the lower treatment rate. So all these phenotypes, all this data, massive work done by the research team, collecting all this information. And then at the end, as the researchers, we got the data, and then we classified the pigs in different groups: resilient, susceptible, and also we considered those animals that died before the end of the experiment. And those that had lower treatment and highest growth rate, then those pigs, they were resilient. So that's how we did the classification.
And that would just be then under typical commercial conditions.
Right.
Who's performing?
Yeah. That was the idea. We didn't want to. What we wanted was to test these pigs under commercial, real life scenario, no? And this was a polymicrobial challenge, which means that we didn't introduce any pathogen artificially. No, we just introduced sick pigs. And this is a common scenario in every farm. No, pigs interact with each other. Some are sick, the others become sick. And then some get treated more, some get treated less. Some recover very quickly their growth, or some they don't.
Yeah, it's interesting, because like you said, this is what I think this is where the science is going, especially when we're talking about reducing antibiotic usage and stuff like that. Is like, well, how do we breed more robust and resilient pigs and feed them to be more resilient, right? So I wish you had a few more correlations with some metabolites, so we could be like, okay, yeah, I got to focus on that one. But
right. Yeah, that's right. It's very important to have, especially when we do selection or the breeding, have a balanced breeding where we balance every aspect of a pig life, let's say.
Yeah, no, very interesting. Okay, so before we get to the final three questions, I hope you've been thinking about them. I'll just ask, like if there's one or two take-home messages that you want today's listeners to get from the episode, what would those be?
Yeah, right. Yes, what I want is to say that research is worth it. That's what I want to say. Yeah, because we maybe quite often we don't get what we want, and we want to get to that. Every good research needs validation, no? So, and that's the holy grail of the research, validate the results. So I think when we do that, and then we have a big breakthrough, no? So I think it's worth it. Keep doing the research.
Yeah, no, definitely. I think it's a good one. And, to tie it back to your feed intake or restriction study, right? If we had just continued to go with the belief that lower feed is going to cause increase in tail biting, you'd never have found that out, right? So it's one of those things that we to avoid even getting stuck in the trap of like, well, we know that, so why would we test that, right? It's always good to go and to do the research anyway. And sometimes you're surprised. And I think those studies are the ones that are the most interesting. They might be the harder ones to write up and publish, but
I know. It rejected my hypothesis.
Oh no, science doesn't like those studies, but those are that's why we do it, is, we're not always going to be right. So,
right.
Okay.
It's time for our famous three.
So I think that'll bring us to the end of the topic part of it. So I'm going to ask the same three questions that we ask all our hosts or our guests on here. The first one is, what would be your favorite go-to, I'll do swine related, or you can do agriculture related or science related resource or book?
Okay. Yeah, I wasn't prepared actually for these questions. There are so many books that I've read, and it's challenging to pick one, no? But as a researcher, I always go back to the research book, to the physiology books. I love physiology books because they help me understand. And despite that I'm at this point, I always go to the physiology book.
Yeah, there's no wrong answer, right? It's what do you go to all the time, and so it's so the next one, maybe this one will be a little bit easier, is what would be a favorite book or something that you would recommend, that you've either read recently or in the past that you'd read over and over again if you could.
Oh, okay. This is easier then. It was just recently I read a book which was kind of interesting to read, and it's called Good Life, if I'm not mistaken. And this is one of those books that make you think that the relationships that we build with each other, they're really important. And this book was based on a Harvard study and it's still going. It's like more than 80 years of following people. It's a research, and they follow people's life, adult life, and the one and only good predictor of a good health when you get old, it's good relationships.
And 80 years to back that up. I mean,
yeah, and still going. They're tracking generations. Yeah, the kids, the nephews, the grandkids. Yeah, it was interesting. Yeah. It's the truth, but it's shocking to hear it, to read it. Yeah, good.
I think part of it is you're thinking, well, it can't be that easy. I guess it's not easy, right? Relationships are not difficult, but it's like, you're expecting something not as simple as that, I guess.
Exactly.
Yeah.
All this sophisticated research, and then you get to something so simple. Good relationship, good friendships, good family relationships, and yeah.
Yeah. No, very interesting.
It was the only predictor for the health.
to look that up.
Sorry, maybe this can be transferred to the pigs. If we provide an environment for good relationships between pigs, maybe they'll have less health issues.
Maybe. I mean, there's another study. You have to put that grant in. Okay, so our last question is, you can either think of it from going back to successful leaders, or you can even go like successful, say, swine producers specifically. What would be a characteristic that you think sets those people apart from maybe some less successful?
That's a great question, Dan. I've been so fortunate to work with my mentor, Dr. Graham Plastow. It's a professor at the U of A. I've been so lucky to work him, and he's been an example of for me to follow, not only the professional part, also as a person. So I think what a good leader is they lead by example, and that was the example that I got during all my career at U of A, having somebody to lead by example and not talking too much, just lead by example. So I think that's a feature that characterize the leaders, and also another feature is they give people the possibility or the opportunity to express themselves. They don't put you in a box. So this is one of those features that Dr. Plastow had too. So yeah, these are the two main.
Yeah, I've definitely seen those listed before as very important for having good teams and being a good leader. Yeah, so no, that's great. Okay, well, that brings us to the end of another episode. So I'll thank you again for coming on and sharing your work with us and some of the findings, and hope that you enjoyed it, and hopefully we'll have you on again in the future.
Yeah, thank you for the opportunity. Of course, I did enjoy it very much. And the last thing that I want to mention was that, and I want to give a deep appreciation for this project that I talked was funded by the government of Alberta, the research-driven agriculture research. So RDAR. And I want to thank them for supporting us with this project. This was, without them, wouldn't have been possible.
Yeah, great. It's always good to acknowledge the ones that are given the money.
Yes, they make our research possible. Yeah, thank you, Dan, so much.
Yeah, no, thank you again.